Okay, we're now recording, and thank you very much, James, for agreeing to give about the cell seminar, and it's all yours.
Thanks so much, Kate. Well, yeah, so I'm James Carothers. I'm an associate professor at the University of Washington in chemical engineering and bioengineering, member of the Molecular Engineering and Sciences Institute and the Center for Synthetic Biology. Before I came to the University of Washington in 2012, I was a postdoc with Jay Kiesling at UC Berkeley, and before that, I earned a PhD with Jack Shostak at Harvard. In my group at the University of Washington, we essentially do three things. We use large-scale computational simulations to interrogate metabolic control system design spaces to try to identify configurations that might increase flux toward some kind of bioproduct that we'd like to make. The second thing that we do is we build CRISPR-Cas transcriptional programs to implement the results of those designs. And then the third thing that we do is engineer programmable aptamer biosensors as genetic regulators and as ways of reading out the functions of the systems that we build. Now, in my presentation today, I'm going to tell you about some new work that we've been doing to build multi-layer CRISPR AI circuits for dynamic genetic programs in cell-free systems as well as in bacteria. And by way of kind of high-level introduction, what we're really thinking here about is whether we can create CRISPR-based tools for programming gene expression to accelerate the development of bio-based production systems. So what I didn't tell you in my kind of brief high-level introduction is that we're really interested in developing these technologies and core approaches for engineering both cells and cell-free systems as platforms for bioproduction, where we're specifically interested in molecules that have either high medical or industrial value. Now, the application of CRISPR tools for transcriptional repression in bacterial metabolic engineering is well established. By comparison, the creation of CRISPR tools for transcriptional activation in bacteria have lagged. So we've been working for a number of years now with our collaborator, Jesse Zalatan here at UW, to develop new tools for bacterial CRISPR activation that by identifying effective transcriptional activators, by systematically defining rules for effective bacterial CRISPR activation, and by creating approaches for encoding functions in engineered guide RNAs. And so we've shown that these CRISPR-8 tools, so CRISPR activation tools, can drive expression of multi-gene biosynthetic pathways from multiple promoters and multiple bacterial hosts, including E. coli, Pseudomonas putida, as well as E. coli-derived cell-free transcription translation extracts. And so what I'm going to do today in this presentation is describe some of our most recent results and outline some strategies for integrating CRISPR activation with CRISPR inhibition to create new capabilities for building multi-gene and dynamic genetic control programs for both cell-free and bacterial systems. Okay, so I always like to start with this kind of big picture view. That is that, in principle, there's a vast space of medically and industrially important chemicals and materials that you can produce using biology. In fact, it's not always appreciated that bio-based chemical production is already a $160 billion industry in the US, and that we're on track for something like 25% of all the chemicals produced to be bio-based by 2025. And there have been a number of notable recent successes in applying synthetic biology and metabolic engineering for applications in bioproduction. But the fact is that these remain challenging, time and resource intensive undertakings. And a lot of this is because the fundamental challenge here is really that you have to program the activities of a large number of gene products, and they all have to function together as a system inside either the cell or the cell-free reaction that you've designed for them. And so, I think it's worth thinking about, what exactly is happening when we're introducing new metabolic functions to a pre-existing network. And of course, a lot of this has to do with the fact that, let's take bacterial systems first. If we introduce new multi-gene functions, so if I want to build a new metabolic pathway to convert some input substrate, like a simple sugar, and turn that into something that has high value, then what I'm really doing is introducing a series of metabolic functions to the system. But underlying or into that system already exist these really complicated metabolic networks. And so, these networks are generally perturbed when we introduce new functions to them. And a lot of times those perturbations happen in unpredictable ways. And it's really because of that underlying network complexity that makes it really hard to figure out what a new perturbation or what a new introduction of function is going to be. So that's kind of at the macro level. But even if you drill down into a specific engineered pathway that you might stitch together by expressing a series of enzymes, there are still going to be these control problems that arise. And I'm going to use an illustration just to kind of make this point. So in my own lab, we've organized a lot of our work around the production of industrial aromatics, including molecules like the ones shown on this slide, p-aminostyrene. So this is a vinyl aromatic whose polymers have tunable optical and mechanical properties that are useful for a number of industrial applications. So we're making PV panels or making polymers for tissue engineering. And what we find is that, in principle, you could make p-aminostyrene, so that's the molecule shown on the right here, from glucose in about 15 steps by introducing 5 heterologous or non-native gene products to a microbial metabolism. But what happens when we do this is that we can see that, and these are what I refer to as control problems. These are the things highlighted in red down there at the bottom of the slide. We can see that there are, even in a short stretch of pathway like this one, there are intermediates that are readily lost from the cell. So your flux would be diverted from the bioproduction pathway that you're trying to build. There are other intermediates that are toxic to the cell at relatively low concentration. So that's like p-aminocinamic acids, PACA. There are other enzymes, if you overexpress them or express them at the wrong time, they have a deleterious effect on the metabolism of the cell because they eat up all the aromatic amino acids in the cell. You could express an efflux pump to get the product out of the cell, but if you do that the wrong way or at too high levels, then you'll destabilize the membranes and reduce the health of the culture that you're trying to engineer, and thus, of course, the amount of product that you get. And so, at a very simple level, we can see that, these are problems that are specific to this pathway, but really this kind of general class of control problems is going to arise in almost any engineered pathway or engineered system that you'd like to construct. Now, in principle, there are dynamic feedback mediated regulatory control systems that could be implemented in a system like this one to solve or to address and mitigate some of these control problems and enable high levels of production. Of course, in practice, now what we're really thinking about is adding an additional layer of complexity on top of the already complicated existing metabolic network and control system network that we need to engineer into a system to enable this to be produced. Okay, so what have we been doing? So our goal really is to create some designable expression tools that we can use to kind of rapidly program the timing and expression levels of large numbers of genes in this system. So really what we want to do is create a set of tools or create a set of capabilities that we can use to rapidly implement multi-gene programs that then we can analyze for the impact on some phenotype that we care about. So we have this idea, the underlying networks are complicated. There's these vast kind of control system design space that we'd like to access. But really what we want to do, or at least the purpose of what I'm going to tell you in this talk is, can we create some new genetic tools that will allow us to implement complex system designs in a way that is straightforward and can be implemented using kind of genetically compact constructs. So here's what we're going to do. So we target genes using nuclease defective dCas9, just like everybody else, with a guide RNA that specifies a target site on the DNA. So if we target this complex to a promoter or an open reading frame, then this is going to give us CRISPR inhibition. If we want to enable simultaneous activation, then we use modified guide RNAs. So we call these scaffold RNAs or scRNAs that include a 3' MS2 hairpin that recruits a transcriptional activator that's fused to an MS2 coat protein. So we can express multiple SG RNAs or scRNAs to inhibit and activate genes simultaneously. And so again, the SG RNA is targeted to a promoter or result in CRISPRai, and scRNA is targeted to an appropriate site upstream of a promoter, give us CRISPRA. And so this relatively simple, kind of straightforward, at least to say conceptually, is the underpinning of the systems that we want to be able to build to implement these multi-gene regulatory control systems. So now let me tell you how we have, or why this story gets a little bit more complicated. So in the first part, I'm going to tell you how we've improved and generalized synthetic gene regulation of bacterial systems. We're not going to wind this all the way back to kind of its start, but kind of jump in at a high level so we can figure out or tell you some things that we've been doing recently. So, so this project really started with this challenge, that is that getting effective transcriptional activation in bacteria had always lagged transcriptional activation systems that have been engineered in eukaryotes because of the differences in the ways that bacteria or prokaryotic systems regulate transcription from their promoters. Namely, if I take a eukaryotic system and I have some transcriptional activator and I bring it within several hundred nucleotides of a transcription start site, then you will generally initiate transcription from that promoter. In bacteria, what we knew, or what the field knew before we started this work, was that the rules or the constraints on activation from bacterial promoters were significantly more complex and harder to understand exactly how you would recruit a transcriptional activator to a particular site and get transcription. So what we did is screened a series of transcriptional activators to develop this new system for CRISPR activation-directed transcriptional activation in bacteria. And so this system gives us the basis for building these kind of fully programmable platforms where we can target some genes for activation and some other genes for inhibition. Now, immediately coming out of that work, we had some questions, some observations from that initial work, through all the trial and error that was kind of necessary to make it work. And so here are some basic and basic questions that we knew we needed to address if we wanted to kind of build this into a full-fledged genetic programming system. So, for example, which promoters can we actually activate? What's the impact of a promoter sequence or the presence of transcriptional repressors? Can we get a clearer view of the CRISPR-A distance dependence that we'd observed? We've seen that you really had to put the transcriptional activator at a very precise distance from the TSS if we wanted to get transcriptional activation in the first place. This is probably why CRISPR activation in prokaryotes had kind of lagged the development of similar tools in eukaryotes. So really what we wanted to understand was how we could most effectively activate genes from synthetic or engineered promoters so that we could control heterologous gene expression. But then we also wanted to develop a set of rules that would allow us to activate genes within bacterial genomes. So, one of the first questions that we asked was whether the intrinsic promoter strength would affect the level of CRISPR activation that could be achieved. And so for these experiments, we're using a test set of minimal promoters that span about a 200-fold range in basal expression levels. So going from the left-hand side from kind of barely detectable to maximum CRISPR activation on the right-hand side, which incidentally, if this means anything to you, is comparable to kind of strong TET-induced expression. And so what we find is that CRISPR activation is sensitive to promoter strength. It's not necessarily surprising. You can't significantly activate genes that already have high levels of basal expression. And then at the other end of the spectrum, at least for these in this test set, we weren't able to significantly activate expression from promoters with the weakest levels of basal expression either. But then there's that kind of broad swath of the Goldilocks middle, where the basal level of expression and our ability to activate it are kind of operating independently of one another. So, so one of the things that we'd seen in our original kind of description of the CRISPR-A system is that there was this really strong, as I said, kind of strong dependence in the distance between the transcription start site and the guide RNA target site within the promoter region. And so now what we've done is we're going to add more resolution to that analysis. And we found something interesting. And that is that there's this really well-ordered rise and then fall and then rise again in activation as we move away from the transcription start site. So that's what's shown on the right-hand side of the graph here. So marking back on the horizontal axis is the distance to the transcription start site. And then the locations there are the target sequence at the end of the guide arc. And so what we can see is that, just as we'd seen before, that there is really an optimal distance here around -81, but we can now see that there's quite a bit of activity that can be recovered at -91, and even a little if you move a little bit closer at -70, and even some if you kind of stretch all the way out there to -102. eventually it falls off completely. And what we assume is happening here is that presumably the CRISPRA complex is kind of rotating around the DNA, and that it needs to be on the same side as the polymerase in order to mediate those interactions between the transcriptional activator, which in this case is success, to the RNA polymerase kind of holoenzyme to get activation from these promoters. So through these experiments and a number of others that I don't have time to present today, kind of a picture of the complexity of the task of activating endogenous promoters emerged. But we nonetheless reason that it might be possible to apply what we've learned to identify targets in the E. coli genome that could plausibly be activated using CRISPR activation. And so for this experiment, what we're doing is selecting a subset of genes with target sites. So the main constraint here is whether they have protospace or adjacent motifs that are the cognate sequence for the particular dCas9 variant we're using. In fact, for these experiments, I'm showing you S-pyrogenase dCas9 and an expanded PAM recognition dCas9 variant. So that's DXCas9 3.7. And then the other thing that we're looking for here, we can use RNA-seq data sets to do this, is to identify promoters where the basal expression levels aren't too high. So we don't want kind of maximal expression where we wouldn't expect to be able to add additional activation on top. And we don't want them to be too low. And then there's kind of an additional constraint, which isn't much of a constraint, and that is that they have to be regulated by sigma 7 so that they be compatible with success. That's something like 90% of the E. coli genome. And so then what we're doing is measuring CRISPR activation from endogenous genes or endogenous promoters, I should say. And what we found is that our predictive rules enabled activation of 4 of the 6 endogenous E. coli promoters that we targeted. Now, of course, there's a lot more that we'd like to understand about the rules for activating endogenous genes, particularly whether we could make CRISPR-A directed expression levels quantitatively predictable. But even at this stage, it looks like something like 80% of the E. coli genome might be targetable using these tools and the general approach. Okay, so I'm going to shift gears just slightly and point out that the work I've described to you, kind of our development of the system, has taken place in E. coli, the kind of model workhorse of every laboratory, right? But there are a number of what you might call non-model microbes that have proven useful for bioproduction applications. And there are a bunch of work underway to develop new microbial hosts that have desirable either metabolic capabilities or that can utilize target feedstocks or that can withstand harsh bioprocessing conditions, that sort of thing. There's a lot of interest in recent years in developing these non-model microbes as bioproduction hosts. I'm most familiar with the US Department of Energy's Agile Biofoundry sponsored kind of set of hosts, many of which are shown in this slide, but there are a number of others. Now, the important thing here for the purposes of this discussion is that the RPOA RNA polymerase gene is well conserved across many different bacterial species, including within the regions that interact with SOCS, which is the transcriptional activator that's been at the heart of a lot of our work. That's what's indicated in the purple here. And so, what we take from this is that it suggested to us that it should be possible to port bacterial CRISPR activation from E. coli into diverse bacterial species. Now, let me just make a clear point about why would we want to do this. So let's go back to our engineered p-aminocinamic acid or p-aminostyrene biosynthesis pathway. And I'll focus your attention on the intermediate here, p-aminocinamic acid, the thing shown in orange. Now, this is a simple experiment. All we're going to do is just add increasing amounts of p-aminocinamic acid. So that'd be a product of this pathway, or one of the final intermediates in this biosynthesis pathway, to cultures that are growing in the presence of E. coli or Pseudomonas putida. So Pseudomonas is on the left-hand side. It's a gram-negative soil bacterium that's gotten a lot of attention recently as a potential chassis for bioproduction because it has these really nice metabolic capabilities and the capacity to survive in kind of these harsh bioprocessing conditions. High reducing power, metabolize a lot of different feedstocks, including the toxic products of aromatic lignin degradation. And it was that last property that kind of made us think, I wonder if the Pseudomonas putida would be able to survive in higher concentrations of some of these toxic aromatic products that we want to make ourselves. And therefore, would this be a better starting point for engineering a system to produce some of these toxic molecules? And so what I'm showing you on this slide is that Pseudomonas is something like 3 times more resistant to this p-aminocinamic acid pathway intermediate than E. coli. So, I'm not going to show you kind of all of our work that we've been doing in Putida. That's another presentation. But I will say that what we have done is implemented CRISPR activation in Putida using the system that we'd previously developed in E. coli. And so to do this, we kind of developed a methodology to transfer CRISPR activation to a no host by first optimizing expression levels of the CRISPRa components themselves, so the transcriptional activators, the guide RNAs, the dCas9, and then applying or systematically investigating the rules for effective CRISPRa that were kind of based on this characterization of synthetic and then endogenous Pseudomonas putida promoters. And so we've seen not only that CRISPRa programs can be implemented, but also that levels of gene expression suitable for metabolic engineering can readily be obtained in Pseudomonas putida using this new set of tools. So, so that gives us now a set of rules for kind of improving and generalizing gene regulation in bacteria. We developed these rules in E. coli, and then we found that they can be applied to other non-model microbes with metabolic capabilities that we're interested in. We're starting to develop CRISPR activation in other systems as well. But now what I want to do is move on to kind of a different set of problems here. And that is, we want to use what we've learned about the rules for doing CRISPR activation in bacteria to design some new CRISPR activation components so that we can build these multi-gene expression programs in bacteria. So, in the first part, I kind of showed you that CRISPRa is sensitive to a number of factors. And what we wanted to do is see whether we could incorporate, kind of turn that into a set of design rules and add some additional sets of constraints. So in this first set of tools that we want to build are, we want to have a set of design rules for kind of no cross activation, no binding sites for known transcription factors, and no homology within a set of target promoters. So these are going to be a set of engineered promoters that we want to be able to activate through the expression of its cognate engineered scRNA. And the reason that we want to do this is so that we can start to build out those engineered heterologous pathways and program different timing and expression levels at each of the different promoters within the system. And the real upside here is that we'd be able to deliver programs that can modulate kind of a new expression program just through the delivery of a set of small guide or small scaffold RNAs. So, build one set of target kind of software or target hardware rather, with maybe a pathway encoding multiple genes, and then operate on that hardware by delivering sets of small guides as the software. So in our first set, we've got 4 synthetic orthogonal promoters. And we can see here that we can get as much as 50-fold activation. So that's terrific. That's more than what we need for all of our kind of metabolic engineering applications. But one of the things that we wondered, though, is, all else being equal, why is it that we don't get 50-fold activation from all the promoters in the set, even though, at least at this point, they were all designed kind of in the same way? And, we'd done everything that we could kind of on the promoter side of things. But then we turned our attention, and this is something that people in the other parts of the CRISPR engineering field had sort of noticed, that RNA folding effects could have profound impacts on the kind of CRISPR-based functions that you get. Now, in some of our own other work, we've seen that kind of co-transcriptional folding trajectories can have a big impact on RNA functions. And a lot of these impacts result from what are best understood as kinetic effects that are governed by the sizes of the energy barriers that separate the structure that an RNA folds into at the end of transcription from the structure that's needed for its function. And so, we wondered whether differences in these spacer sequences and the impacts that those have on folding into active scRNA structures could be responsible for the differences that we observed. So what we did is built a set of something like 40 different promoter scRNA pairs. And what we found, we analyzed a bunch of different kinetic and thermodynamic parameters, but the one that had kind of the controlling or most significant impact on function, most predictive impact on function, is something that we call the kinetic barrier, the folding barrier that separates the guide RNA at the end of a transcriptional process, or its minimal free energy structure, from the structure that's necessary for function. And there's a really strong correspondence here between the effectiveness of scRNA-directed CRISPRA and the predicted kinetics of scRNA folding. So now, if we take our kind of sequence-based promoter design rules and our structure-based guide RNA design rules and put those together, now we can build a new set or design additional components that have, so a promoter and a cognate scRNA that can activate that promoter. And when we do this, now we can build promoters that have the same level of expression as the original one that was the best in the set, so this J3 promoter. And so now we get kind of predictable and tunable and highest levels of expression compared to what we saw with J3. In fact, we've now used this approach to build a set of 8 different orthogonal CRISPR promoters that can all be targeted independently for programmable expression. Now, there's one last capability that we wanted, and that is we want to be able to tune the level of expression without modifying the promoter. So remember I said that we want to encode these pathways as a kind of hardware and then have the scRNA programs operate on them, or the software kind of operate on them. And so, there are a number of approaches in the literature people have taken to kind of engineering guide RNAs to modulate their association with target DNAs. The approach that we've taken is to truncate the 5' end of the spacer sequence. We thought that this would just be a relatively straightforward way of reducing the affinity of the scRNA and the dCas9 complex for the engineered promoter, which would reduce the level of transcriptional activation that we get. And so now what we're doing is applying these sequence and structure-based design rules to not only generate tunable levels of CRISPR activation, to not only generate these orthogonal promoters, but also to generate tunable levels of CRISPR activation. So now we can target these different promoters independently for programmable and tunable expressions. That's what I'm showing you here on the right-hand side. Okay, so now what can we do with these? So the first thing we want to do is, test these synthetic promoter tools. Can we actually use them to program combinatorial variations in gene expression and access these combinatorial expression design spaces? So this is a straightforward experiment where we've put each of 3 different engineered promoters to regulate the expression of a different fluorescent reporter protein, so RFP, BFP, or GFP. And we're going to transform the strains with a different program from this library of scRNA programs that each are expected to give high, medium, or low expression from each of the 3 engineered promoters. So this is a combinatorial set. And what we see is that we only get basal expression in the kind of off-target condition. That's the one at the far right. And then predictable variations in reporter expression that depend on the identity of the scRNA program that's delivered to the strain. So showing that the expression of these multiple genes can be independently targeted and tuned using these multi-guide CRISPRA programs. Okay, so let's do something a little more interesting with them. So, so now what I want to do is show you just one example. We've engineered a number of pathways, kind of using several pathways in our lab using these tools now, but let me give you one illustration. So in this experiment, we're going to use CRISPR activation to independently regulate pathway gene expression in a pathway that we've engineered to produce lacto-N-tetrose, which is a human milk oligosaccharide. Now, just by way of a little brief backstory, I'll make this fast just in the interest of time. We know that breast milk is a good source of nutrition for newborn infants. It has lots of different macronutrients like lipids, fats, proteins, and a series of carbohydrates. But then we also know that there are a bunch of other biologically active components like immunoglobulins, growth hormones, oligosaccharides that play important roles in kind of in infant intestinal homeostasis and immune development. And it's these other bioactive compounds, these human milk oligosaccharides that are actually something like the 3rd most abundant solid component of after lactose and lipids. But these are unconjugated complex glycans that They have functions like prebiotics, anti-adhesives, antimicrobials, but they're notoriously difficult to synthesize at scale using traditional organic chemistry. So that makes them potentially good targets for development of bio-based production systems. So anyway, so now what we're doing here is driving the expression of 3 heterologous genes under the control of the J3, J5, J6 CRISPR activation promoters. So the same promoters in scRNA pairs that I showed you with the fluorescent protein expression. And now what you can see from this, let's call it a focused library, so a subset of all the possible scRNA library designs, we can see that, that we can generate significant amounts of this lacto-M tetrose, so LNT, that's the desired complex, desired compound. But what's really interesting here is that we get this significant LNT production only from the kind of low, high scRNA program, illustrating what is, kind of consistent with a lot of other observations in the field that, generally speaking, just expressing the highest levels of enzymes in multi-gene pathways are rarely give you the highest levels of production through those pathways. And this kind of underscores our interest in developing these tools for rapidly implementing these combinatorial expression programs in the first place. That if we want these tools to be able to interrogate design spaces and identify combinations of expression programs that give us high levels of production, well, this is the kind of tool that we can use. All right. So, so now in what I've just shown you is that we can, kind of use the rules that we uncovered through our kind of systematic characterization of promoters to design CRISPR activation components for multi-gene expression programs in a couple of different bacteria. So now I've shown you in Pseudomonas fluctita and in E. coli, at least one example of an engineered pathway, and we're working on other engineered pathways as well. But now I want to, in the last couple of minutes here, kind of shift our attention to the idea of building dynamic genetic programs in both cell-free and bacterial systems. And before I do that, I want to take a quick step back and make some high-level observations about gene regulatory network engineering. And the first is that developments in gene regulatory network engineering have underpinned a lot of the advances in synthetic biology and metabolic engineering as fields, just in general. The second is that approaches for programming static gene regulatory networks have advanced significantly. It's not too difficult to engineer static gene regulatory networks that have different levels of gene expression that are constant with respect to time. But it's been significantly more difficult to kind of increase the sizes and complexities of dynamic regulatory networks. And this is kind of a funny, simple illustration here, just going from Mike Elowitz's work, kind of at the beginnings of the synthetic biology field, 20 years ago, and building the repressilator, to some recent really nice work. But in some ways, it's sort of a funny way of saying, well, how much progress have we really made here as a field? But kind of joking aside, I think it's been really hard to integrate multiple components. And the strategies have generally not been scalable. And so I think this is really why a lot of the big and kind of increasingly complex systems that we're able to build are typically triggered, but not what you'd call dynamic. And so we're really interested in this idea of whether we can kind of take steps toward building dynamic gene regulatory systems for that we can use to kind of couple with our multi-gene CRISPR AI targeting to increase the sizes and the complexities of the systems that we build. Now, one last thing by way of introduction, there have been a number of successes in recent years toward the kind of bottom-up construction of synthetic cell systems that use cell-free transcription translation, so cell-free expression systems, to recapitulate, but in a lot of ways, reimagine how cell-like behaviors can be constructed. And that, this audience doesn't really need to be told this, but just to kind of remind you that synthetic cell systems employing different boundary systems with different metabolic substrates and activators, different chemical information systems to program functional outputs have been built. And of course, there are a number of efforts underway to develop cell-free expression systems for bioproduction, both in industry, but also in academia. there are some interesting bioproduction phenotypes that we're interested in, and that we're developing systems to produce plant natural products and to convert carbon dioxide into industrial chemicals. But really, what I'm going to tell you in the next in the waning minutes here are just about the dynamic control circuits that we're building, the technologies that we're building that will enable us to do this. So, our first observation was that if we develop approaches to incorporate CRISPR activation with CRISPR inhibition, this would not only increase the number of theoretically realizable network topologies, but also give us a compact control system that we could use to implement complex gene expression dynamics in either cell-free or CFS systems or in cells. And so to go beyond kind of elementary operations of activation and repression, we formalized a framework for constructing these kind of higher order CRISPR AI circuits. And so the way they're going to work is by operating through interconnected CRISPR AI nodes. And in this framework, one of these nodes, these individual nodes, are discrete transcriptional units that contain target sequences for CRISPR activation or CRISPR inhibition directed transcriptional regulation. So then we stitch the nodes together, and that's how you build the network. So, one of our first challenges here was to adapt the CRISPR activation system developed in E. coli for use in cell-free systems. And again, we had these kind of dual purposes. One is we want to develop cell-free as a platform for bioproduction to make some of the molecules we care about, but also we wanted to use this to prototype dynamic regulatory networks that then we could maybe move back into bacteria. So, the first thing that we'd notice is that there was a really good correlation between kind of cell-free and E. coli, but for both basal promoter strength and promoter activatability by CRISPR activation. And so, this really makes us think, I guess the functions are relatively similar across both of these systems. So then, to characterize these individual CRISPR activation and inhibition nodes, we're going to isolate all the CRISPR A and I components onto individual plasmids. So this lets us do kind of independent titration of expression levels for all the components that are in the system. And so what we found, and what I'm showing you, or what I'm not showing you rather, is that the concentration of the scRNA or sgRNA expressing plasmid gives us higher overall levels of activation and repression, as well as faster onset of the control action. So if we want to assemble these individual units to build multi-layer circuits, then we need to build kind of these activation and activation-repression cascades by doing level matching between the input and output dynamic ranges between the sequential nodes. And so what that really means is that we need to be able to match the output transcription levels of an upstream node that encodes an scRNA to the relevant transcriptional input range of a downstream node that encodes another scRNA or sgRNA. And so, what we'd seen based on these kind of plasmid-based titrations, we saw that both CRISPR activation and CRISPR inhibition respond to changes in input guide RNA expression levels that span something like 2 orders of magnitude. And across this responsive range, the CRISPR-A controlled transcriptional output layer levels could vary something like 24-fold. And so, while the dynamic range of CRISPR-A generated outputs does not fully span the dynamic range of the inputs, at least not yet, this characterization does show us that the individual CRISPR-AI nodes can be sequentially combined by carefully matching the kind of upstream output dynamic ranges to the downstream input dynamic ranges to get these layered operations, and that these are relatively predictable. So, once we've got these kind of rules governing the construction of the multi-layered CRISPR AI circuitry, we wanted to build something more complicated, so a little more complex transcriptional programs. And so now what we're going to do is combine a CRISPR activation repression cascade with an additional node of CRISPR activation to form an incoherent type 1 feed-forward loop. So this is that kind of classic pulse generating circuit. And so if we take this kind of level matching into consideration, we can see that the circuit topology determines the timing of the gene expression. That's what's shown in the left-hand side of the figure. But we don't see any difference in kind of expression dynamics between CRISPR-A and CRISPR-A plus I, so the CRISPR, the blue and orange lines there, at low concentrations of sgRNA that are expressing the plasmid. But when CRISPR-I is activated by CRISPR-A to form the IFFL, then we get a gene expression pulse that's qualitatively different from expression that's generated by either one alone. Now, what's really exciting about this is that we can also tune the timing of the gene expression pulse generated by the type 1 IFFL by varying the concentrations of the sgRNA expressing plasmid. And so, what we see here and what I'm kind of showing you in this illustration is that the maximum gene expression, the maximum output rate happens something like 2 hours earlier in a cell-free reaction when we increase the sgRNA expressing plasmid concentration tenfold. And more generally, what we find is that the time of the maximum gene expression pulse can be continuously tuned over something like a 4-fold change in sgRNA plasmid expression levels. So shifting the expression maxima earlier by up to 2 hours compared to unregulated expression. And moreover, we can capture the kind of expression dynamics and of the system using relatively straightforward mechanistic models that at least now give us a framework for engineering these dynamic circuits. We've also shown that we can build orthogonal circuits operating all in the same reaction that don't seem to interfere with one another, up to 7 different guide RNAs. And so this really gives us a way of thinking about how we can have a framework for engineering dynamic circuits in these cell-free systems. Now, the very last piece of data that I want to show you is just that, we started with the E. coli system. We, in developing the CRISPR activation system, we moved that to the cell-free system, used this as a platform for developing those dynamic circuits. And then now the last thing we want to do is show that we can move the system that we've developed in the cell-free system back to E. coli. So in this last experiment, I'm just showing you that we can build CRISPR activation inhibition networks that can function in a bacterium. And so these are kind of 3 proof of concept circuits that we built. In the top, there's kind of CRISPR activation that's going up. And the middle is an activation repression cascade that kind of starts going on and then turns off. And then the last one is this type 1 incoherent feed-forward loop that gives us the characteristic function that we want, which is really a combination of the top 2 curves. So starting low, going high, and then coming back down again. That kind of classic expression pulse that we want. All right, so let me kind of briefly summarize this last portion, and then we'll wrap up here. So what we've seen is that the strength of the CRISPR activation and inhibition can be controlled through guide RNA expression levels. So that is, we can build these circuits that can operate to the regulated expression of guide RNA. So we aren't changing anything else except the guide RNA expression. We can build these multi-layer circuits by doing level matching between the CRISPR-A and I nodes. These multi-layer circuits can encode these kind of different orthogonal and dynamic expression programs. And these dynamic programs can function both in cell-free systems as well as E. coli. And so, we think this is really giving us a scalable framework for doing the rational design of dynamic transcriptional programs in cell-free systems and in bacteria. So, of course, the next challenge, is for us really is to increase the number of components that we have access to, build those components so that they meet kind of our target specs so that we can increase the sizes and the complexities of these systems. All right. So, what I've shown you today is that the requirements for bacterial CRISPR activation, the target sites do pose some challenges, but our data also show that CRISPR-A has the potential to be effective at a lot of different target sites. By systematically defining rules for effective CRISPR-A, we've created some new strategies for improving and generalizing synthetic gene regulation in bacterial systems. We now have a set of tools and kind of emerging capabilities for engineering multi-gene CRISPR-AI programs in bacteria and cell-free systems that are derived from bacteria. So we can encode these programs as genetically compact sets of scRNAs and sgRNAs. These CRISPR-AI programs can specify a lot of different functions. We've already seen that we can activate heterologous genes, we can activate and repress endogenous genes, we can implement dynamic expression programs. I don't mean to give you the idea that this is all solved. We're still learning more about all these systems all the time. But I think these tools are really creating some new opportunities for developing bacteria and cell-free systems as platforms for bioproduction. So I think I'll stop there and take any questions if you have them. And I appreciate, again, the invitation to your seminar series.
Thank you very much, James. I obviously love this talk because I see all the possible applications for building complex synthetic cell circuits. There is a lot of questions in chat. Can you see that?
Let me look. I have not looked yet. I was trying not to. Okay, let's see. Should I just start at the top? Is that the best way to do this?
Yes, if you could read the question too for people that are going to watch the recording.
Sure, yeah. So the first one is, why are eukaryotes easier to control with CRISPR-A, if I heard correctly, compared to prokaryotes? And perhaps related, what's the reason to use MS2 MCP instead of direct fusion of dCas9 with transcriptional activators? That's a question from Lauren Willow. So both great questions. I'll take the first one first. So they're easier to control because there are fewer constraints on the way transcriptional activation happens in eukaryotes compared to prokaryotes. So the eukaryotic transcriptional regulatory machinery happens, largely recruits the polymerase and activating domains just have to interact, can interact across big distances in space. Instead, in prokaryotic systems, the transcriptional regulators that have to sit down next to the -35, -10 region, kind of what in our case might be the SOX box if you're talking about a SOX-S transcriptional regulator, have to sit in particular places next to the promoter, the minimal promoter, in order to get activation. So it's those additional geometric constraints that really make it more difficult to develop these systems. Now, the second part is why to use MS2 instead of direct fusions. And that really is because partly that geometric problem that this gives us a little more flexibility to address the geometric problem, but also because this would allow us, we haven't really done much of this yet, but we could use different activators. We can recruit different transcriptional activators. We can use different orthogonal pairs of RNA and protein interaction to kind of increase the complexity of these systems. So that's really what it's a little forward looking is the idea there. Let's see, speaking of non-model systems you've tried in Cellfree, yeah, I guess that one I answered in the second part. How much do you think we can scale it up, not in different bacteria, but in synthetic cells based on bacteria translation system? I think this means how many CRISPR-A or I targets can we, how big can these programs really be? And I think the answer is we have built systems that regulate something like 6 different heterologous genes. And we've shown that we can combine 3 nodes of CRISPR activation with 1 node of CRISPR inhibition, which isn't exactly the same thing as saying we can rewire an entire network. But other people have shown that you can build arrays of CRISPR-I of say 10 or 15 different genes. And so I think that maybe starts to represent, let's say at least, a good target for us would be, can we get to that 5 or 10 or 15 guide level in the next couple of years? And that would really allow us to test how much bigger can these programs be. Let's see, what's the next one? If you use CRISPR, and TXTL, how do you deal with guide RNA stability in the presence of nucleases? We haven't really seen that this is that guide RNA stability is really much of a constraint in the cell-free systems. I think part of it might be because once the guides have associated with dCas9, then they seem relatively stable to nucleases. But that actually raises another kind of interesting problem or interesting challenge that we have, which is how to reverse one of the CRISPR activation or inhibition events. And especially in a system, in a cell-free system where there is essentially a kind of one-way activation or inhibition event happening. What happens in a cell is that every time there's a cell a division, the DNA polymerase kind of blasts off all the CRISPR complexes. That doesn't happen in a cell-free system. And so we need some strategies, and this is a kind of an outstanding challenge to reverse some activation or inhibition. So I would actually say, instead of having a problem with guide RNA stability, we actually have a problem with that perhaps that the complexes are too stable. So that might be something worth thinking about. Let's see. Are there any computational tools that help you design these types of multi-layer circuits? If so, what are they and how can they be improved? I think these are still early days. We have shown that we can use kind of mechanistic modeling to predict what's going to happen with these different kind of tuning actions. And of course, one of the things that we'd like to do is make that look a little bit more like forward rational designs. I think we have the components in place, but we haven't started doing that kind of work yet. So I think that would be terrific. It would be exciting. Let's see, have you looked into or maybe plan to look into multi-stable systems like toggle switches and toggle, yes. Yes, so one of the things that we really want to do is build a memory unit. So build something that looks something like a bistable condition that can record some sequence of events. And so, our idea is to use something like a toggle switch, but this is still work that we have and that's going on. Follow-up question, can you do Boolean logic with these activators and repressors? Yes, I think the answer is yes. So we've, let's see, 3 or 4 years ago, we did some work with Eric Clavens' group here at the University of Washington to show that we could build kind of these CRISPR inhibition networks in eukaryotic systems that could do Boolean logic and it could really complicated Boolean logic, up to 7 different guides that could be expressed at the same time and build this kind of fancy logic. We haven't started doing this with A and I systems yet. We imagine that the most interesting things to us are the kind of dynamic functions or dynamic capabilities that you can get, but the kind of theoretical space of circuit topologies that you can access if you do A and I gets big pretty fast. So I think that's something that should be coming. Let's see, and the last one is potentially a naive question for someone in the chromatin field. Are people engineering Broadway switches as a means to tune expression based on metabolic state? Could this be implemented in tandem? Yeah. Yeah, no, this is a great question. Yes, the short answer is yes. So the idea of building, let's say, aptamer or riboswitch-based dynamic regulatory controllers is one that we think is pretty exciting, both for kind of as biosensors for reading out production, but also for implementing dynamic feedback controls. So one of our ideas, and I hinted at this, but I didn't really say exactly how we would do this, but is to build aptamer biosensors that target intermediates in these pathways, use those to control the expression of guide RNAs or the expression of the CRISPR activation complex itself, and then use that to implement dynamic control circuits. So we'd like to kind of close the loop there. Part of our lab has been working on developing these aptamer biosensors. Part of our lab has been working on building these CRISPR transcriptional regulatory systems. So we'd really like to put those together, we and some others as well. And the last one, let's see, is can you use other CRISPR proteins? And I think this probably means, could you use Cas12 or other dCas variants, I think, for doing CRISPR activation? I think the short answer is yes. The longer answer is we haven't tried to do this ourselves just because, there have been so many things for us to kind of learn about the kind of core systems that we know about. But I don't really see any reason kind of fundamentally that, we're really just using the deactivated Cas proteins to recruit a transcriptional activator somewhere. And so, if you use a different dCas protein, there might be different rules or different kind of targeting constraints, but I think that's just a matter of doing the experiments. I think the kind of systematic characterization that we've done using these synthetic engineered promoters, I think that kind of approach could be used with an alternate or alternative Cas system, and it should work just fine. I don't see any reason why it wouldn't. All right, well, let's see. Any other last questions or anything I didn't answer satisfactorily, Kate, or what do you think? I was trying to read them fast, but maybe if yeah, I don't know.
I think you answered everything really perfectly. I'm obviously really excited for the multiplexing. I would like to see more and more. I kind of have a question. I don't think you mentioned that, but I'm sorry if you did and I missed that. Can you control whole operons, whole like big sets of genes?
Mm-hmm. Yeah, yes. Well, you didn't miss it in this talk. I think I've talked about this. I've presented some of those data in other talks, but not this one. Yes, the answer is yes. So we usually think of this as controlling transcriptional units, where in a prokaryotic system, a transcriptional unit could consist of, probably as many as 5 different open reading frames, but at least for us in the systems that we work with, we do up to 3 open reading frames. So we're building a system for that p-aminocinamic acid production, where the first 3 genes are a single operon. And we've seen that we can control that through using our CRISPRA system just fine. So in the system that we've engineered there, we have one promoter that controls 3 genes, another promoter that controls 2 genes, and another promoter that controls a 3rd gene. And then you could add on top of that some inhibition of some other target if you wanted to kind of redirect flux to the pathway. So yeah, so no, I think that's, I don't think that there are any constraints, because once you get transcription happening, then it's just transcription in just the same way that you get from any other polymerase system, if that makes sense.
Yeah, it does. Thank you. One of the biggest problems we have is obviously gene expression control if you want to build a complex synthetic cell. And I feel like your work is one of the solutions, because we have to have something that works reliably and doesn't rely on a cell cycle.
Yeah, thanks. No, we're pretty excited about. I mean, we mainly think about this in terms of the bioproduction systems that we build, but what we really want to be able to do is have kind of multi-phase reactions where maybe you express parts of a pathway or parts of a system, maybe even make some phospholipids that could give you a platform for expressing a membrane protein later on that then gives you some product. So this starts to be a relatively sophisticated kind of process control problem. And even though, and so I imagine that same kind of setup or the same problems that we encounter could also be used for the kinds of things that you're talking about. So yeah, I hope so. I think so. We've got a lot of this, the last part of my talk, all that work and this kind of toolbox of components. So we're going to have that available to share soon. The manuscript is under review, and I think we'll go through soon. And so we'll make all those components available to everybody. And I think that could be a good starting point for building the kinds of systems that you're talking about. We're happy to share and I'm happy to help people learn how to use the tools too. I don't know if they're on this call now, but there are a couple of members of my lab who often come to the seminar series. I think I've come to some of the other Build a Cell workshops. I haven't managed to make it myself yet, but I don't know. I guess if our pandemic goes away or something, then maybe I can do it. So yeah, I think it'd be fun. You
guys were on a short list for our next workshop location before the world went down the drain.
Yes, I remember that. I remember talking about that. Yeah, exactly. Well, we'll see.
One day.
One day, exactly. One foot in front of the other. We'll see where we get, right? No, this is great. All right. Well, I appreciate, again, the invitation, and I'm happy to answer questions if anybody wants to send me an email, or I can connect you with people in my group who did the work and stuff. So this is great.
All right. Thank you very much. Thank you so much.
Thanks, everybody.
For the talk. Bye, everybody.
Thanks, Kate. Thanks, everybody.